Hi, today my partner María Clara Ortiz and I, Manuela Ochoa, are going to present the article named In Vitro Induction and In Vivo Engraftment of Kidney Organoids derived from human pluripotent stem cells.
So, the stem cells are the cells which all other cells that have specialized functions are generated. So, the stem cell divides and gives some new stem cells or daughter cells that these ones will turn into tissue or organs.
Chronic kidney disease, especially end-stage renal disease, can threaten the life of the humans. There are two options for treatment. The first is blood dialysis, but this can affect seriously the quality of life of the humans. And the other one is organ donation, but the organ donation cannot supply all the clinical needs. There's other option that is 3D renal structures, but it has two disadvantages. The first one is the nutrient supply, and the other one is the low degree differentiation.
Determining whether the microenvironment of renal capsule in immunodeficient mouse undergoing unilateral nephrectomy could promote vascularization and differentiation of kidney organoids.
Methods: reverse transcription quantitative or RT-qPCR. It is used to identify the expression of genes using a necessary material mRNA that is transcribed into complementary DNA by the reverse transcriptase. It is need to have a control group, that in this case is GAPDH, that doesn't have the enzyme and is considered the housekeeping gene. In this study was used to identify the mRNA levels of T, TBX6, and LHX1 that are molecular markers for primitive streaking. Also for GATA3, HOXD11, AJA1 that are markers for mesoderm. Western blotting. This is a technique used to identify proteins by their molecular weight. It is based on a reaction between antigen and antibody and uses head electrophoresis to separate the fragments of genetic material by size. In this study was used to identify the markers for renal podocyte, POD-XL, and also the NHPS1 and WT1 to confirm the previous results.
Some of the other methods were immunofluorescence analysis of 3D organoids. So, the idea with this was to choose an antigen, then they put an antibody, and if the antibody complex form, you should see with the fluorescent microscope something like this. So, you should see it green, red, or blue, and in this article they use like LTL, ECAT, and DAC. So, hematoxylin and eosinostaining. So, it's composed in two stages. The first one is a nuclear staining by a basic stain that is the hematoxylin. It will have affinity for the acid that is in the nucleus. So it will look like this, like purple, sometimes blue. And the other stage is the eosin that is an acid stain, and it will have affinity for the cytoplasms and it will stain pink, like you can see it here. So they use this to see the morphology of the kidney organoids.
Results. During the induction process, the cellular morphology changed a lot. At the beginning, the cells express a large shape, like you can see here. So in the study, they changed CHIRC99-021 to FGF9, and this helped the cells to express a fuller shape, like you can see here. Immunofluorescence. We have here the markers, and we have the day of differentiation when they were evaluated. So we have T and LHS1 that are markers for primitive streak. We also have TAPI, that is a marker that binds strongly to adenine and thymine, and we have MERGE when we have both of them. So we can see that the day where they have a major expression was the day 4 of differentiation. At MERGE, here we can see that both T and TAPI had an equal expression, but here in MERGE, we can see that the TAPI had a major expression than LHX1.
The results of fluorescence that were for the antigens GATA3 and WT1 that are mesoderm markers. So in day 0, there were antigens for each one of them. So, and in the TAPI they are positive because it has a strong union for adenine and thymine sequences in the DNA. In the 7th day, the organoid became to grow a little, so they had a little expression of GATA3 and WT1. So they were positive, and that's the reason you can see the line here. Merge means that they merge the two of the fluorescence inks. Okay, the present result here shows that the kidney organoids that have been transplanted gain volume compared to the ones before the transplantation. It means that the kidney organoids were getting nutrients from the host. So, here in this result we have the LTLT and the ECAD. There are tubular markers. So, the LTLT has more fluorescence, it means that it is positive and it has more antigen than this one that you can see a little red, but it's not so fluorescent, but it still have antigen for it. And here we can see the Windsor blotting, that it means the red and the WT11 that are irritable markers. So here and they get PhD, that it means that this one is for control. So the red, you cannot see it in day 0 to 7, but in day 12 it has a little expression here. You can see it over here. And the WT11, it expresses in the whole time from day 0 to day 12, but day 12 has more expression.
So here at the image D we have the expression of the collective tube markers that are GATA3, ECAD, and DAPI at the kidney organoids. We also have the image E where we have the expression of the podocyte markers also in the kidney organoids. Western blotting. Here we have the podocyte markers and we have the group control. We can see that the second marker, that is WT1, began to be expressed at the day 7 and the other ones were highly expressed at the day 18. This result suggests that the podocyte cells like may exist at the kidney organoids.
Here they are evaluating again the tubular markers. So you can see here the ECAD, the LTL, and DAPI. Here, remember, the DAPI are it gets to you with the adenine and the thymine. And here you can see in the merge it results like the LTLT, and you can see a little bit the red because there is not too much of ECAD in it.
Here at image F, we have the detection of the NPHS1, that is a marker for podocytes. We also have the LTL and the DAPI markers. At merge, that is when we have all of them, we can see that the one with a major expression was the LTL. At image G, we have the marker CD31, that is a marker for vascular endothelial cells. We also have LTL and DAPI. We can see at the merge that the one that has a major expression were CD31 and DAPI. Discussion. Here we have some authors and we have their studies. We also have if what they demonstrate was achieved in this study or not. So first we have Vanderbilt. So he demonstrated the generation of vascularized human kidney tissue derived from the hips that are human-induced pluripotent stem cells upon transplantation of human kidney organs to renal capsule in mice, and this was demonstrated in this article. We also have Dilworth. So he demonstrated that left nephrectomy, which was performed on a host rat, improved the growth of transplant metanephroid. That was also achieved. We also have Hammerman. So he said, identify the renal primordia metanephroid transplanted into animal renal capsule undergo organogenesis in situ. They become vascularized by blood vessels of host origin and exhibit excretory function. This was not achieved in this article or this study because the kidney organoids didn't express an excretory function.
So in conclusion, we had the molecular biology allows to identify the expression of different cell markers that can help in research and medicine studies. The second one is that some of the cellular processes that occur in the organisms are well known as described thanks to the molecular biology. And the third one is that molecular biology has helped in the development of better laboratory techniques in order to get more precise results to guide the personnel to better diagnosis, and also helps to get new treatments.